So welcome everyone. The speaker today is Lida Kanari from EPFL, and the title of her talk is A Topological Understanding of Neuronal Morphologies from Single Cells to Detailed Neuronal Networks.
Thank you, and thank you for inviting me to give a talk at this very interesting series of seminars. So I will talk about neuronal morphologies and how we can go from single cells to networks and using primarily topology. So first, an outline of the talk. I will start by explaining what are neuronal morphologies and why they're important. Then I will describe briefly the topological morphology descriptor and some applications. Then I will discuss computational synthesis, so how can we computationally generate neurons? And then I'll give some insights on how we can understand this process through topology. So let's start from the basic neuroscience. So why is it important to study neuronal morphologies? On the left, you see an image of an actual biological mouse brain that has been sliced, and you can see all the small neurons and the intricate structures in this brain. What we want to do in the Blue Brain project is to generate a computational model that includes as much as possible all the details that we see in biology. So starting from the positions of the cells and including increasingly larger amounts of information to go deeper into the biological structures. And here is an idea of what we want to include in the models. So ion channels are small channels on the surfaces of neurons that control the molecules that go into the cells. Morphologies define the shapes of the neurons, and electrophysiology is if you stimulate these neurons with electrical currents, what would be the response of single neurons? And all these are at the level of single neuron. Then at the level of networks, we have brain regions. So the brain can be separated into different regions. If we go deeper into each one of these regions, we see neuronal networks that form between the neurons, and they generate the neuronal activity that we observe. So this is already a quite complicated picture, but we are not including a lot of the structures that are present in the brain. So there are other cells, such as astrocytes and microglia, that are not directly active. And that's why they are not considered that they are creating the neuronal activity that we see, but they influence neuronal activity strongly. So I'm going to give an example later on of microglia and how we can use the techniques that we are currently using for neurons on other brain cells as well. But what are neuronal morphologies? So at the beginning of 20th century, Ramon y Cajal, who's considered the father of the modern neuroscience, he was going into the microscope and drawing these shapes of the neurons by hand. And he started observing that they have these very complex branching shapes. So his hypothesis was that all these all these branching structure of neurons give rise to networks which are controlling the functionality of the brain. And this is actually confirmed to be quite true with recent technologies. As you can see here on the left, we have examples of branching structures of different groups of neurons within the brain of rodent. So all this is the same within the same species, either from different brain regions or from the same brain region. And you can see that they express a very high variety of shapes. Similarly to botanical trees that have a high variety of different branching structures, the neurons express many different branching shapes, and the hypothesis is that these branching shapes define the functional roles of the neurons. So to some extent, this is currently confirmed. So we can say that depending on the shape of the neuron, we have different functionality on them, but it's not a straightforward relation. So it's very hard to pinpoint exactly which parts of the neuron contribute to the different functional activities that we observe in the brain. So why do we need to study this neuronal shape? First of all, as I mentioned, they form the foundation for the electrical behavior of the neurons. But in addition to that, they constrain heavily the connectivity of the brain networks. So depending on the different branching shapes, they can connect differently, and they can form different types of networks. So to summarize, they overall influence the dynamical properties of the brain networks. That's why it's very important to understand the neuronal shapes and what's the difference between them. How can we describe neurons topologically? So here is an example of two very different neurons, a Purkinje cell and a pyramidal cell. They're both from a mouse brain. But as you can see, they look very different. And when you look at them, you can say immediately that, okay, this is more dense, it's more flat. The other one has more sparse branches. So the differences are apparent. But what about this example? So these are pyramidal cells from the mouse brain, and they look pretty similar to the naive non-expert eye. So that was actually the motivation for me to try to develop topological techniques to understand neurons, because I couldn't see the differences that was apparent for the experts. So let's start from the topological morphology descriptor, which I'm going to call TMD. So this is pretty much the algorithm to transform a dendritic tree into a persistence barcode. And the idea is to encode each one of the branches within the tree structure as a bar, where the first point of the bar corresponds to the start of the branch and the last point of the bar corresponds to the end of the branch. And the persistence is preserved, so we start from the longest to the shortest bars. The persistence barcode can be alternatively represented as persistence diagram or a persistence image in the two-dimensional space. And these two representations are usually preferable because they're easier to use as inputs into classifiers or into computational tools to compare them. TMD is stable with respect to a few perturbations on the input tree. And these are errors that could happen on the specific positions of the nodes. So as you have each point within the tree structure, we have tested what's the impact if you just move them by epsilon. And the second perturbation is if we add or subtract small branches. So to both of these type of perturbations, the TMD has proved to be stable, which means that if we take a tree and an associated function, let's imagine for now that this function is the radial distance from the soma. And if we slightly perturb the tree in terms of the perturbations I mentioned before, by epsilon, then we see that the bottleneck distance between the TMD of the original tree and the TMD of the perturbed tree is bounded by 3 epsilon. So this means that we have a stable descriptor of the neuronal structures. Now, if we go back to the previous example, here is the persistence image of a Purkinje cell and the persistence image of a pyramidal cell. And as you can see, they're very, very different. But if we go to the more difficult, the more challenging example, we see that this neuron, which is named by the experts as tufted pyramidal cell, has a second high density of branches further away from the soma, which in neurobiology, it's called a tuft. And the other neuron does not have this tuft present. So that's why the experts name one untufted and the other tufted cell. So now if you look at the persistence images, it's quite clear what's the difference between them. But what's interesting is if we subtract the two persistence images, so we see the difference, the clear difference of the tufts, but we also see a secondary effect of branches close to the soma. So this tree is not only having a second high density of branches further away from the soma, but also has longer and more branches close to the soma. So this way we can use topology to understand exactly what are the differences between two types of neurons. So now we'll use TMD for a few applications. The first one will be classification of pyramidal cells. So for this application, we had the pyramidal cells classified of rat classified into specific groups, and we were trying to see how well the TMD could discriminate between these groups. Here you can see an example of layer 5 cortical layer pyramidal cells. So this is a deeper layer in the cortex. And there we have these very big tufted pyramidal cells, which have a very clear and nice second tuft of branches. Then we have the tufted pyramidal cells with a smaller branch, with a smaller tuft, which indicates smaller and fewer branches on the tuft. And then we have the third category, which does not have a well-defined tuft at all. And all these types are very well picked up by the topological descriptor. So we use this technique to validate how well the experts group each cell type. And we found out, so these are pyramidal cells from all the cortical layers of rat. And we found out that the TMD could perform very well. So it had a very high accuracy into discriminating each one of these pyramidal cell types. And here is just a nice visualization of the types that you've seen before. So we start from superficial layers in the cortex, and as we go deeper, we see we have larger pyramidal cells, and the topological descriptor could properly describe all these different shapes. And now we will go to one more challenging example, and that's clustering of cells. And here in this data set, we collaborated with Idan Segev and Christian de Kock. Idan is from Israel and Christian is from Amsterdam. And they gave me human pyramidal cells. You can see some examples here. But the challenge was that the experts could not separate them into classes. So what I did is I just extracted the persistence images of the two, of all the cells. And I found two distinct classes. One was expressing a tuft, similarly to the rodent data, and the other one had an absence of tuft because you had very sparser branches in the tree structure. And interestingly, so these are examples of the more representative cells for the two classes. But what was interesting is when I tried to classify the 100 cells that I received from the experimentalists, I could classify them very robustly into the two classes. So only 5% of the cells were not clearly divided into one of these classes, but the rest of them were nicely falling into one of these categories. And what's more interesting here, which links back to the property I mentioned, is the functional properties of the cells. So for this data set, we also had functional properties. So we had information on how quickly these cells fire. And when we separated them into these two classes, the experimentalists also tried to see if there is a functional difference between these two classes. And indeed, they found out that there is a significant difference in how fast these cells spark. So the profuse tufted are much faster, and the slim tufted are much slower. And that's a very distinct functional property that we observe here. The catch is that we still don't really know why. So we can associate the shapes with specific functional properties, but we haven't figured out what's the biological mechanism that generates these different patterns. The final application I'll talk about is the microglia. So with a group in IST Austria, the group of Sandra Sigert and with Catherine Hess, we collaborated on this project. They had a very nice data set of microglia from different areas of the mouse brain, and they wanted to distinguish them. So this is an idea of the pipeline. It's slightly modified compared to what we did for the neurons. So again, we extract the persistence barcode, the diagram, and the image. But microglia tend to change very quickly. So because they respond to they have immune responses to inside the brain, what they do is they change branching patterns quite frequently in time. So we can't no longer consider them stable as neurons are. And for this reason, we observed a very high variability in the shapes, even within one brain region. In order to solve this problem, we created averages of persistence images. So we grouped 10 or 50 cells of microglia, and we generated an average image. And these average images generated very distinct patterns for each one of these brain regions that we had in the mouse brain. So if we use UMAP and we group the microglia of different brain regions, we see a clear separation between them. So some general observations, persistence images have been very useful to represent biological data, but they're averages. So I'll show you an example how these can be misleading. And even though we have proved stability of the topological descriptor with respect to common topological distances, they might not be the most relevant distances to use in the case of neurons. So what would be a good way to compare barcodes of brain cells? Here is the problem. So here I have one persistence diagram of 5 points. And in the second one, I added some points close to the soma, and the third one, I added points far away from the soma. So if we take a look at the first and the second one, we would assume that the difference is in the tufts, but that's not the case. And again, with the third one, if we take a closer look, we might be confused and assume that there are less branches in this structure close to the soma. The problem here is that densities are defined with respect to the number of points within the same diagram. So this might create a confusion on where the differences are localized. And if we try to measure distances between these three diagrams, bottleneck will have no difference detection because the persistence module is the same, so it's stable. So we wouldn't be able to recognize them as different diagrams. And the Wasser's time, because these points are close to the diagonal, again, it won't have a very small difference between these diagrams. But okay, so these points are pretty small, so are they really important biologically? So here's an example of of a student in Blue Rain working, Francesca working with Alexis and Maria in my group. So what they did is they took a neuron and they tried to see what would be the effect if they changed the location of two branches within the tree structure. So they simulated these trees and they found that there is a huge voltage change within the two structures, even though the change in the underlying tree is pretty small. So that's an example of why small branches or points close to the diagonal do matter for the biology. So how can we solve this problem? What's an appropriate distance that we can use? Perhaps the problem lies in the concept of distance. So maybe a single value number is not sufficient to describe the observed differences into the biological structures. So maybe we need to extend this definition of distances to find more appropriate uses of differences. An idea would be to define statistical differences between diagrams. So here we have the three previous examples, A, B, C. B has more points close to the soma, and C has more points close to the on the tuft. In order to identify the differences, what I did, I put a circle around each one of the points on the persistence diagram, and then I compute if this circle is covered in each of the diagrams. So if you have points between the diagrams that you're comparing that are present, let's say both in A and in B. And if you do not have these points present, then we add one of these difference points. So here we identify which points in one diagram are not represented in the others. So if we take B and we do this process with comparison to A, we found out that there are points close to the zero, so close to the soma, that are not present on A. Similarly, if we take C and we subtract A, we found points that are further away from the soma and they're not present in A. And a similar difference between B and C. So if we now go and create the persistence diagrams of the differences between the persistence diagrams ABC, we see that we identify the differences between them very well. So we can capture these differences. A different idea would be to create Gaussian kernels. So if we try to approximate the point distributions with Gaussians, we can identify the means of these Gaussian kernels. And this gives us a better understanding of larger differences. So again, if we put these Gaussians, we see a small shift in the positions between the diagrams, but we don't see a huge difference. So this is an indication we can capture significant differences between the diagrams with this method, but not small changes. And another idea comes from the method developed by Wojciech, Martina, and Jens in KTH, and they developed the stable ranks. So this is like an integral of the points on the persistence diagram. And what you can do is you can use different rank functions to create these curves. And we use this technique in the example of the microglia in order to create the UMAP of the stable ranks. And as you can see, the important point here is that we could separate the brain regions pretty well using stable ranks. So this is an indication that we can still identify differences using stable ranks that were not obvious with common topological descriptors. So now that we have understood a little bit how this works and how we can go from the trees to the world of the barcodes, how can we go back? So once we have the persistence barcodes, how can we use the inverse problem to create trees that are biologically relevant? So the idea is pretty simple. We will start from the soma of the neuron, and we will start growing in something like a random walk. So each point is added after the other, and we have a probability to bifurcate and a probability to terminate. And that's where the topology comes in. So the probability to bifurcate at the distance d from the root is defined exponentially by the birth time of a bar in the barcode. So each time we have a new bar, the probability of a branch to bifurcate increases exponentially. And each time in the barcode, a bar dies, the probability to terminate increases. So this way, if we follow from zero, from the soma of the neuron the bar, we see that the probabilities to bifurcate starts increasing when we see a new bar, and then we have a bifurcation. And then the probability to terminate the bar increases when we have the death of one of the persistence points in the underlying structure. So if we use this and a much more complicated algorithm, we can generate cells that resemble to the biological ones. So here is an example of this process. We take a biological reconstruction, we get the barcode from it, and then we can synthesize one cell that looks similar to the original cell. And then we can use this method to generate many different examples of the same cell type. So here are the different pyramidal cells, one example from each cortical layer that we've seen before. And we have synthesized all the different types within the cortical layers of rat. And we see that we get a structure similar to what we would get under the microscope, what would we observe in a biological cortex. Of course, the similarity in the appearance is not a validation, so we need to properly validate the computationally generated cells. And we do that with various steps. So the first step is to take a long list of morphometrics. And morphometrics can be measuring the total length, the angles, the number of branches that we generate. And we compare these statistics with the biological statistics that we extract from reconstructions. So here we have some of the features presented, but the list of features we checked is quite long. So we tested around 200 features, and they all agree between the reconstructed and the synthesized cells. The second test is to try to simulate these neurons. So we use a computational model which puts some current on the soma of these neurons and generates some electrical traces. And we compare these electrical traces if they have been generated using the biological reconstructions and if they have been generated using the synthesized cells. So you see, they might look a little bit different in terms of structures, but they generate the same signals statistically. So we can confirm that not only they look similar, but they also behave similarly in terms of electrical properties. But are we really generating the same neurons? And what would be a definition of how similar are the neurons that we get from biology to the neurons that we computationally generate? So trying to understand that with Adelie and Catherine, we tried to develop a framework in which to describe this process. So how we go from the barcodes to the trees, and then extracting the barcodes again, and then comparing the original barcodes to the synthesized barcodes. And then similarly, if we go from the tree, so we start from an original tree, we use a barcode, and then we synthesize it. How different is the original tree from the tree that is computationally generated? And here is a representation of a barcode, and then how we can see where we need to put one of the bars. So this in this work, we focus mainly on the combinatorics of these properties. So the idea is, if we have this bar, how many trees can we generate from this bar and from this barcode? And this is just the red bar. It can be attached to different positions within the tree because we need to respect the elder rule. So it can only be attached to bars that are smaller than it. And in this particular example, you see that there are many possible locations in which you can attach this bar. So what happens here is, if we take the bar, we can the barcode, we can order each bar depending on the birth time, and then we can assign a permutation from that on the death times. So the death times are practically a permutation of the birth times in the underlying structure. And each barcode can be associated with a number of possible trees that it can realize. And I'll go back to that in the next slide. But the idea is that if you change the order of the bars, so let's say we start from this order, which would correspond to this one. So if we start changing one of the bars and increasing its length and up to the point that it changes order with the second bar, let's say, we go from one representation to the other. And here we are not taking into account at all the length of these bars. So we are not concerned with how long there are the bars. We just concerned about the order of the bars in the barcode. So here is an example of all the possible orders that we can have and their permutations. So for this example of this barcode, they're permutations of S3 So that's what we call S3 equivalent barcode. And the idea is, as we change each one of these bars, so as we go from one to the other representation, it's like a circle. So we can, by changing the lengths or changing the order of the bars, generate all the different possible combinations. And here is a very nice representation done by Adeli, where we can see all these possible combinations, their equivalents to the symmetric group, and the trees that can be realized. So all the possible combinatorial trees that we can generate based on each one of these barcodes. And you see that the number of trees that we can generate can differ quite a lot. So it goes from 1 in the case where all the bars can only be attached to the longest one, and it goes up to 6 in this particular example when each bar is included to the previous one. So in order to count how many trees can be generated from each bar, what we can do is we can use the product of the indices of the bars. So the index is how many bars each bar is included in. And what we see here is that the set of barcodes can be partitioned into equivalence classes, which are indexed by permutations in the symmetric group. So this is interesting because it defines how many different trees we can generate, and it also defines what is the order in the branching structure and in the barcode. And why is this biologically relevant? So the number of tree realizations seems to be following a very distinct distribution in the biological trees. So if we plot the number of branches versus the number of tree realizations, the black curve is the maximum number of tree realization for each number of branches. And here we have biological trees from very, very different neurons. So here, just a few examples of neurons, and you can have neurons from C. elegans, which are very, very simple, neurons from different types of flies, or the pyramidal cells that we've seen before. And they all fall into the same distribution. And I forgot to put this diagram here, but if you try to generate barcodes randomly, you would see that the tree realization number is much higher compared to the biological tree realization number that we get. So there seems to be an underlying relation in the biological trees that is respected, which we're still trying to figure out what is this like golden rule for neuronal trees. And now, how is this interesting for the networks of neurons? So we have all these very different shapes of neurons, and they seem to have different functional roles, but they still seem to follow some topological rules. The idea, as I started in the beginning, is to try to generate artificial models of the brain. And you've seen before that we could generate a very small portion of the brain, which was called a cortical column, and it has different layers. And now we can generate full brain region. So this is the neocortex of a mouse, and this has been fully synthesized, and you see that's similar like length scale. So this part that we generated before is just a tiny bit of the whole neocortex, and neocortex is just one of the brain regions. But why is this interesting in the global picture? So, okay, we can generate the brain regions, but what we can do with them? So a very interesting application is to try to understand disease models. So it has been observed in experiments that if you have some specific mental disorders, such as stress disorders or schizophrenia, the shapes of the individual neurons change significantly, and it depends where you are in the brain. So where you are in the brain will define how these shapes of the neurons change. So here what I did is I tried to just implement very simple changes on the original neurons to generate different sets of barcodes and see what would be the impact of these differences in the underlying networks. And here are just two examples. So in this example, I just cutted branches. So you keep the same longer structures of them, more persistent components remain the same, but the smaller components disappear. So the smaller branches are stripped out. And as you can see, the network compared to the original network that we generated does not change that much. So the underlying structure of the network remains similar. But then we implemented another transformation, and that was shrinking of all the parts together. So all the branches together. So that meant that the barcodes that we generated were shrunk. So when we do that, obviously the cells that we generate are much smaller as we shrink them. And what was surprising was that the network was very, very fast collapsing in this type of transformation. And that might not seem very interesting because, of course, you're shrinking the neurons, so you expect that they will not connect so much with other neurons. But what is interesting here is that this is the type of behavior that has been associated with more severe brain disorders. So they have observed that neurons shrink in some severe brain disorders, and we are hypothesizing that this could be causing the collapse of the connectivity that is represented in the phenotypes of various brain disorders. And this would be the next steps of what we would like to study in the future. So with that, I would like to thank a lot of people that contributed into these different projects. And first of all, Henry Markram and the Blue Brain project that generated all the biological insights. And of course, Catherine has been my main collaborator with for most of these projects. And then you can see here a very long list of people that contributed on the different parts of the project. And thank you everyone for your attention.
Thank you. So now I would invite everyone to unmute themselves and thank our speaker. And now it's time for questions. Yeah, so feel free to unmute yourself and ask.
All right, I have a question. So in one of your slides, I don't remember what was the number of the slide. You were talking about how the usual distance fail to give the similarity between the objects that you want. Can we go back to that slide? I didn't understand why the distance fails.
It's probably this one.
Yeah. So you were mentioning that the usual notion of distance doesn't give you the similarity that you want.
Yes, well, it depends which similarity you're looking for. So bottleneck, for example, is trying to detect what's the difference between the persistence component, so the major component in the barcodes. And here you would not be able to detect that because the major component is pretty much the same, and what is changing are the smaller components.
I see.
And then with the Wasserstein distance, it depends how far away you are from the diagonal. So because these points are close to the diagonal, again, you would only see very small differences in the Wasserstein.
So with respect to bottleneck distance and Wasserstein, these diagrams are pretty close, but in reality, they represent something that are not close.
Yes, and this is very particular to the biological problem because in reality, they are close. So they're just some branches that are different within the tree structures that they could represent. But for this particular problem, so for neurons, this could have a significant impact on the connectivity or on the functional properties. And that's why it's important.
Thank you.
More questions?
I have a question. So, Lida, this is sort of a silly question, but this golden rule for neuronal trees. Have you checked? Is there any possibility that trees that are plants, also satisfy that golden rule?
That would be great, but I do not have a data set of plants. I would be eager to test this. It would be interesting if they would fall on the same rule.
And then another question that I had, for persistence images, I was curious, how do you choose various parameters, or does it seem that those choices are not so critical for this task? And for some of your other summaries, is it correct to say some of the summaries that you produce, there are fewer parameters that need to be chosen?
So in the persistence images that I generate, I use the default parameters of the Gaussian kernels. I'm not trying to play with these because obviously they could have an impact. But I think in the original paper of persistence images, they indicated that there is a range of parameters up to which the differences are not significant on the persistence images. So I'm just respecting this range and I'm not changing the parameters at all. The other question was with respect to the stable ranks, I would assume.
Yeah.
Okay, so the stable ranks, yes, there is a very important parameter, which is the function you choose. And you can use different functions, but this is one of the strengths of this method, that depending on the function you use to sum up these bars, you can get a different curve. And the advantage here is we start with something very simple, like a straight line, so then you don't have any impact. But then if you would imagine that you have a slide where you could see, like in the barcode, which bars would be more strongly represented, so which bars you would get weight more. And then if you see a difference in the stable ranks, that's an indication that this part of the barcode is important to separate two barcodes. So I'm not doing this work. This is in collaboration with Boche's group and Martina's group, but so they could tell you much more information on that. But that's a general idea that depending on the choice of the function, you can get information on what is the distinctive feature between two barcodes or two groups of barcodes.
Thanks.
I guess I can ask another question if you don't mind. Can you explain again, how did you go back from barcode to trees? The process of getting a tree out of a barcode?
So this one?
Yes.
So from trees to barcodes or from barcodes to trees?
From
Sorry.
barcodes to trees. I guess you call it TNS.
TMD. Sorry. So this part?
Let me see. Yeah, this one.
Okay, so the idea is to start from the tips of the trees and you follow them backwards. Every time you have a bifurcation, you compare the lengths in terms of some distance. So here you can imagine it's a radial distance from the soma. So it's like a comparison between 2 and 3. 2 is longer in terms of radial distances. So 2 survives and it gets to continue towards the root, and 3 here terminates. So this is generating this smaller bar 3, which goes from the tip where we saw this branch first up to the bifurcation. Now, number 2 continues back to here, and then it's compared against number 4, which is smaller, so it continues further back. So 2 starts here, and then it terminates. So this is the number 2 that follows all the way up to here, where it meets a longer component. So 2 terminates here, and 1 is the persistence component that goes all the way from the first time we saw number 1 all the way to the soma of the neuron. So it would be the infinite component in the barcode.
I guess, thank you. But I guess my question was the opposite process, from a barcode to a tree.
Okay. So it's kind of the opposite idea. So you start from the longer bars. So here, this part. So you start from the longer bar. You start growing, and at the beginning you only have one branch that is growing out of the soma. And then you have probability to bifurcate and probability to terminate. At each point you check, like, should I terminate or should I bifurcate? And that's a random factor. And you compare it against the probability. And this probability is exponentially increasing as you see new bars. So the probability to bifurcate increases as you see a bar in the barcode, right? So here, so as you're growing here, as you're approaching this bar, your probability to bifurcate increases and then becomes 1. So if you pass this point, then you should certainly bifurcate. Does this clarify a little bit the process?
Yeah, thank you.
Can I ask a quick question? Okay, thank you. Thank you very much for a great talk. My question is about encoding from trees to barcode. So you said when you encode one single tree to barcode, yeah, my question is, how did you measure the radial distance of the tree? Because the distance should vary depending on the position of the neuron and how each neuron is placed. And so, yeah, I'm just curious. How did you determine the distance?
Yes, so the distance is always measured for the biological problems with respect to the soma. That's how you have invariance. So it doesn't matter where the neuron is or how it's rotated. The radial distance is always with respect to the soma, and which remains invariant.
Yeah, yeah. But distance is biologically measured one, right?
No, the distance is measured from the digital tree. So we have the digital reconstructions, which means we have the exact positions of the cell. But we're not taking biological features. We are measuring from the digital reconstruction.
Ah, okay, I see. Thank you.
Sure.
Any further questions? Okay, so if there are no other questions, then we're gonna stop the recording here, and feel free to continue once we're offline. And Lida, thanks again for your wonderful talk.
Thank you for inviting me.